If you're into exercise and nutrition, at some point, you've heard about autophagy, including some wild-ass claims, like it's the key to anti-aging, or it's a secret cellular process that's gonna unlock longevity, and shit like that. Some would even say that you should train and eat in a particular way in order to activate autophagy, that you should train in a fasted state, or train glycogen depleted from a low carbohydrate diet, that you should engage in some sort of intermittent fasting, time-restricted feeding protocol in order to activate autophagy. Now, in my estimation, they're kind of missing the point and mistaking mechanisms for outcomes, which are the things that actually matter. So in this video, we're going to talk about autophagy and its relationship with exercise and nutrition, its relationship with health and aging, and whether or not targeting autophagy is actually necessary. Now, the first thing I'm going to say is autophagy is important. It is crucial for cellular function and homeostasis. It is involved with maintaining chronic and cardiometabolic health, and it is involved with some of the positive effects of exercise and certain dietary practices. I have studied autophagy. I've written about it, done research on it, so I'm keenly aware of the relationship and impact or potential impact that autophagy has on these things. I'm also aware of the misapplication and overextending of autophagy as it relates to practical applications for people to actually employ in their own lives. On top of that, I'm aware of the exaggeration that people online and social media engage in when talking about autophagy and its impact, particularly on health and aging. For example, people say that autophagy will reverse aging or trick your body into aging backwards. Imagine gaslighting yourself so hard you do it at the level of the gene. That ain't gonna happen, not by autophagy or anything else for that matter. If that actually were the case, that autophagy were to reverse the aging process or cause your body to age backwards, there would have been a Nobel Prize handed out for that shit already, including to a bunch of people on YouTube and Instagram and TikTok. And as far as I know, that ain't happened yet. That doesn't mean to say that autophagy is not important and it's not involved with some of the positive health benefits of exercise and certain dietary practices. It is, which we'll talk about. However, as is conveyed and communicated online and social media, is a very, very far cry from what the actual data and current body of research actually suggest. And we'll get to a lot of that specific data. However, suffice it to say that right now, there's a lot that we don't know. We don't know the actual direct impact autophagy has on aging. We especially don't know the direct impact that autophagy has on aging and health as it relates to exercise and nutrition. We know that it is involved. We know that it's associated with, but there are a lot of things that we don't know. The current state of the literature, which has increased over the last 10 years or so, is unclear on a lot of things. Many questions still remain. And related to the point on research, when we talk about autophagy, the actual research and data that actually mean anything are data in humans. Why? Because a lot of times people on social media are talking about the effect of autophagy in humans, as well as application of that research in humans. And while rodent data and cell culture data are very, very interesting, they can tell us a lot about mechanisms of action, and causal relationships between things in the cells and the tissues, as well as potential physiological and clinical impacts, as in the case in rodents. That has very, very little translatability into humans. And with the human data, there are not enough to come up with any sort of definitive autophagy prescription. And so anyone on Instagram or on TikTok or YouTube talking about you should fast XYZ number of hours and only eat from this time to this time, or you should train in this specific way, They're just misunderstanding and misapplying the data. Although it sounds super sexy and sciencey, that's actually the reason why they're saying those things. Not because they understand the data, not because they understand autophagy, no, because it's going to get you to click or buy their shit. And now I say this as someone who has studied and published about the impact or potential impact of autophagy on health, including age-related health, including how exercise and nutrition are involved with that. So I understand that there are important implications here. However, I also understand that there is a lot of context and limitations that go unspoken about when people talk about autophagy online and social media or whatever. And people might consume that and come away with the wrong idea, with the wrong priorities, with the wrong goals. Now, I'm going to touch on these points later, but let's take a little step back and talk about autophagy itself, what it is though. I want to start by saying there are going to be some things that are left out. There are going to be some things that are touched upon, just kind of glossed over, but not gone into in any sort of exhaustive, extended way. That is just the nature of this type of content. But if you want more on these topics in greater detail or greater discussion on them, then you can check out some of these papers here, some of which I have written. Although I want to caveat by saying not everyone can understand a research paper or the implications of it, or its limitations, because they may not have a background in that topic. That's like me reading a research paper of like 99% of all science. Like me reading a research paper that's outside of my domain. I may be able to read the paper, I may be able to understand the words, but I don't know what it means. I don't know what the implications are, and I don't know what the flaws and limitations are to consider when reading this paper. So I can come away with a mistaken understanding based on my naive reading of a research paper that I don't necessarily have a background in. And this can cause problems. And so while do your own research sounds nice and sounds empowering, it doesn't actually acknowledge that most people are not going to be in a position to really accurately and rigorously evaluate the data. And people online can take advantage of that to push their content or their product. And so my goal as someone who has a background in this area is to communicate and explain autophagy in a way that it is understandable, but actually reflective of what the data actually say. That's my goal. That's my task. We'll see how good I do. So if you don't check out those papers, that's okay. But you can think of autophagy as a cellular recycling system, a system that helps maintain the intracellular environment. And what do I mean by that? Well, in the cell, there's a lot of stuff. There are a lot of things. Proteins, organelles, mitochondria, and like a bunch of other stuff. But autophagy is involved with maintaining the quality of those cellular components. Because if they're not maintained, that could disrupt cellular function and therefore tissue function, and potentially lead to pathological states, disease. If these proteins, organelles, and mitochondria are not maintained and become dysfunctional or damaged, that's going to cause problems for the cell. And so when things are damaged or dysfunctional or accumulating, autophagy goes and degrades those cellular components. What happens is autophagy forms vesicles around the problematic content, and then they fuse to the lysosome where it is then degraded. Now, not only are these items degraded, but their constituents, the things that they're made up of, are actually released back into the cell, where they can be used for other cellular processes, metabolism, energy production, and even protein synthesis or anabolism. And why is that important? Well, if these damaged and dysfunctional items accumulate in the cell, again, that's going to disrupt function. And in fact, many chronic diseases are characterized by an accumulation of these cellular components. And this is the case for aging as well. In addition to that, as we age, autophagy function tends to decline, leading to the accumulation and aggregation of cellular proteins, organelles, and dysfunctional mitochondria, for example. And one point on the mitochondrial aspect, mitochondrial dysfunction is central to a number of chronic diseases, like a lot of them. If there are dysfunctional mitochondria, there are going to be problems, and that potentially could lead to disease. Many diseases have mitochondrial dysfunction. For example, in type 2 diabetes and insulin resistance, mitochondrial function is key in that pathophysiology. And this is the case with a number of other diseases as well as aging. So autophagy is involved with preventing the accumulation of these dysfunctional mitochondria, and therefore allowing properly functioning mitochondria to populate the cell and contribute to normal energy production rather than dysfunctional mitochondria, which can produce things like oxidative stress and inflammation. And this is where exercise and, for example, calorie restriction come into play. And why is this? Well, exercise is a stress. At the level of the cell, exercise is going to impose a number of stressors that are going to function as signals that the cell then has to deal with. And autophagy is a stress responsive system. So therefore, autophagy is going to be upregulated in order to deal with that stress. And I say upregulate because autophagy is always on. A lot of people online will say, turn on autophagy, activate autophagy. Autophagy is always on. The question is, do exercise and calorie restriction affect the relative rates of autophagy? And what are the effects of that? It does seem that exercise at a certain point does increase autophagy activity. And there's a nice body of literature now suggesting that autophagy is involved with the acute responses to exercise, as well as some of the positive health benefits of exercise. Similarly, calorie restriction, which we'll define later because that's important, does also involve autophagy in its health-related effects. However, when you get to the human data, it's a little bit more complicated than that. For example, are the positive health effects of caloric restriction due to some cellular process, cellular mechanism like autophagy, or is it the fact that caloric restriction or caloric control just prevents individuals from gaining fat mass and becoming overweight and obese, which then is going to improve their health and prevent disease? We're going to talk about that. But if we're talking about exercise and calorie restriction stimulating autophagy, why would that be the case? Why would those things stimulate autophagy? It has to do with energy. And in exercise, this is especially the case for skeletal muscle, which is one of the most important tissues in overall cardiometabolic health. So just focusing in skeletal muscle for now. But when you exercise, you are placing a tremendous energy stress onto the muscle. You are requiring lots of ATP to support that muscular contraction, to support that activity. And in response to that, we've got our three bioenergetic systems: phosphagen system, glycolytic system, and the oxidative phosphorylation system. Okay, as it relates to the glycolytic and oxidative phosphorylation system, those systems use carbohydrate and lipids, fat. Specifically, glycolysis uses carbohydrate, whereas oxidative phosphorylation can use both carbohydrate and fat. Now, what autophagy does in response to this exercise stress where there is high ATP demand, autophagy, in addition to degrading proteins and organelles and mitochondria, autophagy can go and break down cellular stores of carbohydrate and lipids, breaking down glycogen into glucose and breaking down lipids into fatty acids, therefore providing substrates for ATP production. So it serves as a system to provide substrates for energy production, which is needed during periods of energetic stress, like in exercise. And similarly, with caloric restriction, that too is an energetic stress. However, instead of like with exercise, where there is a need for more energy, for more ATP, caloric restriction reduces the amount of energy available to the cell. And so just like with exercise, autophagy will go work to release glucose and fatty acids to be used for metabolism and energy production. And so when people talk about autophagy being self-eating, this is what they're referring to. So we talked about carbohydrate, we talked about fats. Now what about the proteins? Can those be degraded and be used for cellular processes? And the answer is yeah. So when these proteins are degraded, again, they're degraded in the lysosome. And these proteins are broken down into their constituent amino acids, because that's what proteins are, right? And in doing so, the amino acids accumulate in the lysosome. And it is this accumulation of amino acids in the lysosome that actually serves as a signal for protein synthesis or anabolism. So like many of you probably know that when we eat some sort of protein, steak or eggs or In-N-Out or something, that does have an anabolic response. Now that protein gets digested, broken down into the amino acids, and the amino acids get into the bloodstream, get into the cells and the tissues, and then get uptaken into the lysosome. And again, it is the accumulation of amino acids in the lysosome that causes the signal for protein synthesis, specifically increasing the activity of mTOR, the mammalian target rapamycin. That is what causes protein synthesis. That is what causes anabolism. And yeah, if we're talking about skeletal muscle protein synthesis, getting anabolic, bro. So there's a relationship between this protein degradation system and protein synthesis that likely contributes to maintaining the cellular proteome or the cellular proteins. Now, emphasis on likely, because we don't actually know. Now, this is a perfect example of how you need to be careful in terms of how you interpret certain types of data, because someone could hear what I just said about autophagy relating to mTOR and protein synthesis, and then come away with, oh, autophagy is anabolic. Autophagy stimulates muscle protein synthesis. And someone can even extend that into like resistance training and hypertrophy, which that is a whole other topic of autophagy and resistance training and hypertrophy and its role in it. But you can't say autophagy is anabolic in humans, because the data that I just referenced came from cells in a petri dish and not in live humans. So you can't say autophagy is anabolic and should be targeted for hypertrophy or something like that. This right here illustrates how certain types of data could be completely misunderstood and misrepresented. The data that I just spoke about came from cells, not in live humans. So mechanistically, yes, there is a relationship between autophagy and mTOR and protein synthesis. There is some linkage between those systems and those cellular processes. However, that doesn't tell you anything about how that plays out in humans, about how or even if that occurs in humans. All that tells you is that mechanistically, there is a relationship there. There's like a bajillion things going on inside of the human body, like all of the time, that for damn sure, cells in a petri dish ain't gonna replicate. The conditions in cell culture are entirely different than the conditions of cells inside of the body. So just because you see something in cell culture does not mean that's what happens in cells in the human. In fact, most of the time, that like never happens. One of the advantages of in vitro cell culture research is, yeah, you can control things to a much greater degree. So you can get a better understanding of, for example, X on Y. You change X, you add X, you manipulate X, and then you look at the effect on Y. Because those conditions are so controlled, if you make a change to X, well, there's way less confounding factors that are going to affect Y. So cell culture research, which I've done a little bit of, is great for understanding mechanisms and mechanisms of action for a lot of different things and a lot of different applications like pharmaceutical and drug development. But in the live human body, it ain't just X and Y. There's like a trillion alphabets going on all at the same time. So be careful of how you interpret and generalize and translate different types of research, which we're going to talk about. Now, I mentioned mTOR, and we're going to talk a little bit more about it later, but a lot of people in the lifespan and longevity space, they don't like mTOR. They think mTOR is pro-aging, that mTOR activation or increasing mTOR activity is going to increase aging, increase the aging process, that you should actively try to avoid increasing mTOR. One of the things that gets brought up when talking about lifespan and longevity and health is the fact that intermittent fasting and calorie restriction increases autophagy and decreases mTOR and therefore is anti-aging. And it's one of the reasons that you should do intermittent fasting. However, like we just discussed, autophagy might actually work to increase mTOR activity. And if that were the case in humans, would that then make autophagy a bad thing because it increases mTOR activity? You can't just operate under the assumption that mTOR is bad. There's a difference between acute, temporary, transient increases in mTOR in specific tissues, like in the case of resistance training or eating protein, versus chronic, dysregulated, upregulated mTOR activity, that which you would see in pathological disease states. Which, yes, dysregulated mTOR has been implicated in some chronic diseases and in aging. But those two things are very, very different. And just to say, full stop, that you should actively try to keep mTOR activity inhibited is just, again, misunderstanding and misapplying the data. That would suggest that you should not resistance train, which resistance training is one of the best things you could possibly do for your overall health and yes, your age-related health. Not resistance training would be one of the most pro-aging things you could do. If you want to have a terrible quality of life as you age and die sooner, don't resistance train. Now, there are other signals that act to increase autophagy activity from exercise and calorie restriction, including calcium signaling and redox state, which again, if you want more on that, you can check out those papers that I mentioned. Ultimately, what autophagy does is clean up the cell, prevent dysfunctional things from accumulating and aggregating and disrupting function. But if we're talking about autophagy and it being upregulated in response to energetic stress to go break things down for fuel, well, how does that relate to the maintaining of the intracellular environment? Because breaking things down for fuel doesn't necessarily translate to improving and maintaining the cellular organelles and proteins and mitochondria by their degradation. So does the energy-related autophagy affect the quality control aspect of autophagy? And yeah, it seems to. There's a nice recent paper that talked about this relationship. The relationship between the autophagy that's used to provide substrates for energy production, or metabolic autophagy as they called it, and the autophagy that targets the cell's mitochondria, organelles, and proteins, the quality control autophagy. And it does seem that as autophagy increases to go break things down for fuel, for energy production, that also increases autophagy and the autophagy machinery that goes to the cellular proteins, organelles, and mitochondria. So the autophagy that is used to provide energy for the cell also increases the autophagy that maintains the intracellular environment, and in so doing, contributing to the quality control aspect of autophagy, likely leading to improved function, and yes, likely leading to improved health. So then how do things that people can actually do, like exercise and calorie restriction and intermittent fasting, how do those things affect autophagy? And do those things impact health and age-related health? And if so, should autophagy then be targeted? In addition to autophagy playing a role in the positive exercise benefits, potentially through by maintaining the intracellular environment and preventing the aggregation and accumulation of deleterious items in the cell, autophagy appears to be involved in some of the normal responses, the acute responses to exercise, meaning autophagy may be necessary for some of the things that happen to the body during exercise. There are rodent data, which again has limitations, and you cannot directly translate that into humans, meaning what happens in a mouse does not necessarily mean this is how it happens in a human. But if you block exercise-induced autophagy, this inhibits exercise-induced glucose uptake and insulin sensitivity, as well as AMPK activation. Now, that does not mean to say that this is exactly what happens in humans. You cannot say that because this was in rodents. And chronically, long-term, if exercise-induced autophagy is blocked, that prevents the protective effects of exercise from a high-fat diet. It actually prevented the exercise improvements in insulin sensitivity and blood lipids. There are also data to show that autophagy is involved with the angiogenesis, so the making of new blood vessels, induced by exercise, as well as the mitochondrial biogenesis, the making of new healthy mitochondria from exercise. And again, to reiterate, these are animal data, not human data. So again, you cannot say this is exactly what happens in humans. You can't say that. But it does give us some indication that autophagy is involved with some of these things. But that's about all you can say. Now, as it relates to calorie restriction or calorie reduction, which intermittent fasting and time-restricted feeding get brought in when talking about calorie restriction and autophagy. But yes, it does seem that calorie restriction does upregulate autophagy. Now, what are the specifics and what are the caveats? Well, if you look online and on social media, you ain't gonna find any specifics or caveats. What you'll get instead is a bunch of crazy claims about calorie restriction or intermittent fasting, that these things are the secret to unlocking longevity and turning your biological clock back. Now, I'll just say, yes, I know that calorie restriction and intermittent fasting or time restricted feeding or 1 meal a day, OMAD, or whatever the acronym is, I know that these things are not the same thing. You can do intermittent fasting without doing caloric restriction. However, sometimes these things kind of get glossed over, either intentionally or ignorantly. I don't know which one is worse. Or sometimes these things are focused on, like super focused on, to the degree that people are arguing and fighting with each other, and you're either team calorie restriction without fasting, or you're team intermittent fasting without calorie restriction because you think intermittent fasting is some magical laws of thermodynamically defined thing that has nothing to do with calories. And you want to let everyone know that you do intermittent fasting. I don't know why people make how they eat their identity, but if anyone were to, I could, because I'm Mexican. I don't think this is enough. In any case, let's talk about calorie restriction and its effect on longevity and lifespan. And as it happens, calorie restriction is the most robust, non-genetic, non-pharmacological method of lifespan extension. If you're a fruit fly, or a yeast, or a nematode, or even a rat, if you're one of these organisms, calorie restriction is a miracle. And if you're one of these organisms watching this video right now, I got questions. Calorie restriction in these simple organisms has a big impact on their lifespan. So for example, in E. coli or fruit fly or yeast, calorie restriction can extend the lifespan of those organisms by 2 or 3 times. But when you move up in complexity, like to mammals, say mice, you're looking at between a 30 to 50% increase in lifespan. Now that's still pretty significant, but it is definitely less than what you see in the simple organisms. And that's not just specific to calorie restriction, but that's with a lot of things. Something can have a big impact in these simple organisms, but as you move up in complexity, those effects get moderated. And as you move up even further to, say, non-human primates, what you see with long-term calorie restriction data, which there's not that much of, is not an extension of lifespan, but a reduction of age-related diseases. And that's what you see in the human data. Of the very few human data of long-term calorie restriction, you see a reduction in the prevalence of many age-related diseases like type 2 diabetes, some cancers, some neurodegenerative diseases, things like that. But what you're not seeing is humans living to 150 to 100 years old. Like, that just ain't gonna happen. Now, it'll help you live longer in the sense of you are less likely to die earlier from these diseases, but you're not stopping or reversing the aging process. And that's not to downplay the apparent health benefits of reduced prevalence and risk of disease. That is as important as anything. You'll live longer because you'll not die sooner, but it's not going to, let's say, extend the maximum human lifespan. It might help you get there, but again, it's not going to allow someone to live to 150, 200 years old. And so when we see claims like calorie restriction or intermittent fasting is going to reverse or stop the aging process, our biology don't work like that. And aging is a part of our biology. And you can thank evolution for that one. From an evolutionary perspective, we're just temporary somatic meat vehicles for our genes that have an expiration date. Aging is not something that we're going to be able to get around at least anytime soon, despite what that one rich guy is doing.
If somebody actually had a way to reverse biological aging, what you would see is that up until the point that a person used this intervention, maybe it's a drug, maybe it's a gene therapy, maybe it's something else, up until the point where they used this intervention to reverse aging, they would age normally. Their biological age would more or less track with their chronological age. Then they take this intervention, and over presumably a short period of time, let's say a year, their biological age would drop quite dramatically. In this particular graphic, it's a 20-year drop in biological age over a 1-year period of chronological age. And then when they stopped the intervention, they would immediately go back to their old rate of biological aging and be parallel to the line of the population or people who had never taken the intervention. And of course, you could imagine all sorts of variation on this. Maybe you repeat the age reversal intervention at 20-year periods, whatever. The point here is that we don't have anything that actually appears to do this. We don't have any interventions that actually seem to make an old person or even an old animal, this has never been done in the laboratory, biologically younger again, where all of the age-related traits are in fact reversed or reduced by some significant amount of biological age. So I would argue pretty strongly that reversing aging is not yet possible. Maybe it will be someday. I don't think it's going to happen anytime soon. What we can do is reverse a subset of phenotypes or traits of aging, things that change with age, some of them can be reversed. We can do that with things that are maybe a little bit more physiologically relevant for biological age, though, like body composition, right? Of course, we all know it's possible to gain lean mass or lose adipose, lose fat mass. Age-related changes in body composition can be reversed. Did that reverse your entire biological aging? No, of course it didn't. We can reverse functional aspects of fitness, VO2 max. We can reverse age-related changes in vaccine response. And we can reverse some molecular changes of aging. None of these things are reversing all of biological aging, which is why I, again, pretty strongly say that nobody has reversed biological aging as an entire process. Now, again, some people are trying to claim they've reversed aging. I would just say they haven't. I would further suggest that the burden of proof is really on them to prove they can make an old animal young again. Remarkable claims require remarkable evidence. So my advice would be, don't believe it until you see it.
We did not evolve to maximize lifespan. We did not evolve to maintain function through our lifespan. We evolved to maximize reproductive success. And once we get beyond our prime reproductive years, our function begins to decline. Our capacity begins to decline because our genes actually don't care about us. They care about getting past into the future. And we kind of get tooted and booted. But there are things that we can do to attenuate the loss in function as we age and maintain and even improve our health as we age. Things like exercise and calorie restriction, or at least calorie control, which we'll talk about, and intermittent fasting, maybe. Now, before we get to intermittent fasting, just a little bit more on calorie restriction, like some practical applications that I think people should consider. When we talk about calorie restriction, as it is defined in the literature, which I think is important so we actually know what we're talking about, is calorie restriction is defined as a reduction in calories ranging between 25 to 30% of previous calorie intake. Now, that's not a trivial amount, but it's not severe enough to cause malnutrition. And one thing to keep in mind, again, from a practical perspective, is when is a calorie deficit no longer a calorie deficit? What do I mean by that? What I mean is when someone reduces their calories, so let's say by 500 calories per day, they're in a 500 calorie deficit if everything else stays the same. However, everything else does not stay the same. You actually have metabolic adaptations, such as this thing called adaptive thermogenesis, where when someone is in a caloric deficit, either through reducing calorie intake or even increasing energy calorie expenditure, there is a reduction in metabolism. There's a reduction in total daily energy expenditure, including basal metabolic rate, including exercise-induced energy expenditure, and just general physical activity induced energy expenditure. So your actual metabolic rate, your overall daily energy expenditure decreases. So that initial 500 calorie deficit is no longer a deficit because your metabolism has essentially adjusted to that new intake. Now, that's a whole video in and of itself that I'll probably get to at some point. But when that happens, you're no longer restricting calories. That is your new sort of energy balance set point. So then in order to get the positive benefits of calorie restriction, do you need to reduce your calories again and again and again and again? Because at some point, you're gonna get to zero. And that ain't good. And in the long-term calorie restriction data, that's not actually what they did. A lot of it comes from this calorie restriction society where they practice caloric control, and they compared that group to individuals that did not limit their calorie intake. And so when you say, look at the anthropometric data or body composition, yeah, they weighed less, they had less fat because they ate less. And how does that translate to the markers of health that they took? Well, if you don't have excess fat tissue, if you're not overweight or obese, you're going to be healthier. You're going to live longer. You will have less disease because excess adipose tissue, excess fat mass is, yes, detrimental for health. So these individuals in the calorie restriction society are consuming, some of the data I've seen, between 30 all the way up to 45% less calories than the controls that they're being compared to. And what would that lead to? Less fat mass, no obesity, no overweightness. And it really seems the case to me that the differences in overall health when comparing calorie restricted individuals versus non-calorie restricted individuals has to do with the fat mass associated with these two groups. And when you look at the samples from the Calorie Restriction Society, many of these people are like in single digit body fat percentages, and they're in their 50s. So if you compare individuals in their 50s that are like 7 to 10% body fat compared to individuals that are not that lean, they have more fat mass in the 20s, maybe even upper 20s from some of the data I've seen. Those differences in adipose tissue and fat mass likely explain the differences in the health outcomes between these two groups. Being single digit body fat in your 50s is going to have a tremendous impact on your health. So the calorie restriction is just a means of which to yield a healthy body composition, to prevent weight gain, to prevent obesity and overweightness, to stay lean and maintain healthy body fat levels. But that's not what you're going to hear on online and social media. There is no magical or profound or biohack of calorie restriction that promotes health. It's just a means of which to maintain healthy body fat levels, which, yes, fat gain, obesity, overweightness does increase your risk for a number of chronic diseases and premature death, thereby reducing longevity. But calorie restriction is not a magical, profound biohack, and that does not sound as sexy. All right, what about intermittent fasting? Intermittent fasting is the thing that always gets brought up in the space of longevity and lifespan and optimal, maximal, whatever. And autophagy is like at the center of the lifespan extending effects and like super focused on and praised, and like people love autophagy. People even got shirts talking about feeling autophagy. Motherfucker, you can't feel your lysosomes. But does intermittent fasting increase autophagy? Yeah. It seems to. However, again, what are the specifics and what are the caveats? And again, if you look online, you ain't gonna see any. And when you look at the research, there's very few human data. And the data that do exist have limitations, which, you know, all studies have limitations. That's not to discredit the current data that are out there, but what you have to do is take each individual study with its limitations as well as its strengths, but within the context of the rest of the body of literature. You can't hyper-focus on a single study and think it's the end-all be-all explanation on a given topic, because one single study can't answer everything. Each study is very, very narrow in terms of what question it is actually answering. The conditions, the experiment, the subjects, these things are all very, very specific, and you have to be specific to understand what effect a particular intervention or treatment may have. However, that causes challenges when trying to extrapolate and generalize to a broader population using different treatments or interventions that are not exactly what are used in that study. And this comes down to the tension between internal and external validity. If you want to know the effect of some intervention or some treatment, you have to control for very, very many things. You have to make things very, very specific. However, that's not how things really are in real life, right? Things aren't controlled. Things aren't consistent. Things aren't the same. Humans living out in the real world are not humans in an exercise physiology lab, or rats in a cage, or cells in a petri dish. And you can't really mimic the real world in a lab. And trying to mimic the real world introduces a bunch of confounding variables, a bunch of confounding factors that can then take away the ability of a study or experiment being able to identify the actual effects that are specific to that treatment or intervention. So you always have to consider what is the internal validity versus the external validity. What is the level of control versus the level of generalizability? And you really can't have both. You kind of have to pick your poison when you're running a study. And when you're reading a study, you have to keep those things in mind. You can't just read something at face value, take that and then employ, let's say, in your own personal practice. So what do the data actually say? Well, 4 days of time-restricted feeding, or TRF, to use the acronym, there's a lot of them, but 4 days of an 18-hour fasting period, 6-hour feeding window, during a calorie maintenance energy intake, so that was good. Calories were controlled, they were maintained, not in a calorie deficit or calorie surplus, they were maintained, showed that, yeah, there were some autophagy markers that did increase in overweight, healthy adults. However, the autophagy markers that they measured was mRNA, not proteins. And it was done in whole blood. And herein lies one of the limitations of this study. mRNA does not actually cause changes in function. So an increase in mRNA of autophagy-related proteins will not cause any changes in autophagy itself. That would require an increase in the proteins, not the mRNA of those proteins. And if you all remember Biology 101, mRNA is the precursor to proteins. They're the code for those proteins, which then get translated and then actually go do things and cause function in the cell. Now, one could say, oh, well, there's mRNA, so therefore there's going to be proteins later, right? Those proteins will be translated from the mRNA. Not necessarily. Not all mRNA gets translated into proteins. There's regulation that occurs at the mRNA level. Therefore, you can have an increase in mRNA of a particular gene, but not necessarily have an increase in that protein. So to look at this particular paper and say time-restricted feeding causes an increase in autophagy, you can't say that because they didn't measure proteins. They measured mRNA. And again, the mRNA don't actually do anything. They don't actually affect function. It's the proteins that affect function, of which they did not measure. And they measured in whole blood, which that's fine, but that's different than autophagy in muscle or heart or the pancreas, or the liver, or adipose tissue, or any of the other tissues that are brought up when talking about autophagy and longevity. Now, some still may take this and say, well, this is a compelling case that time-restricted feeding increases autophagy, and therefore is good for longevity. However, in the same study, they saw an increase in mTOR, which again, for those in the longevity, anti-aging, biohacking, whatever area, mTOR is seen as a bad thing. It's seen as contributing and or causing aging. And the idea would be do things that inhibit and reduce mTOR activity. Now, I'm not saying that. mTOR is neither an all good thing or an all bad thing. It's a little bit more complicated than that, which pretty much everything in biology is. And like I said earlier, there's a difference between an acute transient increase in mTOR in specific tissues versus a chronic dysregulated mTOR activity. Dysregulated mTOR activity in cancer cells is different than acute transient increases in mTOR in skeletal muscle from resistance training or acute protein ingestion. So mTOR bad, it's not that simple. But if you were one of these people that ascribe to the idea that mTOR is pro-aging and should be inhibited in order to promote longevity, then I don't know what you do with this data, because yes, they did show that autophagy markers, mRNA, did increase, but they also showed that mTOR increased. Using that framework of autophagy good, mTOR bad, you can't look at the study and say time-restricted feeding is good for health because it increased autophagy, that was only mRNA, because at the same time it also increased mTOR, again, mRNA. So I don't know what you do with this study using that framework. But sticking with the autophagy part of this study, what you can actually say from this study is pretty limited because, again, they looked at autophagy mRNA, not the actual protein, which the protein is the thing that actually causes function in the cell. It's the thing that actually causes things to happen. And again, it was also done in whole blood. We're going to come back to what increases in autophagy markers actually mean, or what we think they mean. And a second study using Ramadan fasting of 17 to 19 hours for 2 and 4 weeks also showed an increase in autophagy markers in young healthy males. However, this was also mRNA as well as whole blood. And this study was also not calorie controlled. So with these data, for what they're worth, show that time-restricted feeding, which is, you're fasting for a number of hours in a single day, does have the potential to increase autophagy. Because again, we didn't actually see any increases in autophagy proteins, just the mRNA. So to say that it increases autophagy, you can't really say that because again, it's just the code for those autophagy related proteins. But it may. We just don't have specific data on that. Now, what about fasting for a longer period of time, like 24, 36, 72 hours? Now, I don't know who'd want to do that, but I believe this is what is referred to specifically as intermittent fasting, which, if I'm getting some of the terminology wrong, whatever. But what are the data on longer periods of fasting? Longer than a couple hours in a single day. Now, there's very few data on this, but one study showed 36 hours of fasting caused some changes in autophagy-related proteins in the skeletal muscle of untrained male subjects. However, not trained subjects. So specifically, in the untrained subjects, they saw a decrement in autophagy protein LC3-1 and another version of it, LC3-2. However, they did not see a change in the LC3 2-to-1 ratio, which is often used as an indicator of autophagy activity. And they also saw a decrease in P62 protein, with some of these changes occurring as early as 12 and 24 hours. So why would these proteins that represent autophagy activity, why would they decrease after 36 hours of fasting? Now, this is where it gets a little bit messy, because increased autophagy activity can either increase or decrease certain proteins, and to get a clear sense of what is actually happening is very, very difficult. So increases in autophagy can occur several different ways. That can occur transcriptionally, so certain proteins are increased, their expression is increased, as well as post-translationally, meaning the proteins are modified, post-translational modification. So for LC3, there are several forms of LC3. So first, there can be an increase in the translation or the expression of pro-LC3, and then that gets modified and converted into LC3-1, which then gets modified into LC3-2, which makes things complicated. For example, during increased autophagy, there can be an increased amount of LC3-1. Also during increased autophagy activity, there is an increase in the conversion of LC3-1 to LC3-2. It gets lipidated. It gets converted LC3-1 to LC3-2. Therefore, if we just look at LC3-1, during both instances where there is increased autophagy activity, that can be either reflected by an increased content of LC3-1 or a decrement in LC3-1 content because there is greater conversion from 1 to 2. That's just LC3-1. What about LC3-2? All right, so there's a greater conversion of LC3-1 to LC3-2. However, during increased autophagy activity, LC3-2 is also degraded at the lysosome. It's part of the vesicle, and it gets degraded at the lysosome. So therefore, when we're looking at LC3-2, increased autophagy activity can be reflected or indicated or cause an increase in LC3-2 content because there's, again, greater lipidation and conversion from 1 to 2. However, it can also cause a reduction in LC3-2 content because it gets degraded at the lysosome. So autophagy can hypothetically be indicated by either an increase in LC3-1 or a decrease in LC3-1, or an increase in LC3-2 or a decrease in LC3-2. Or if we're looking at the ratio, which is often used, an increase in LC3 2-to-1 ratio or a decrease in LC3 2-to-1 ratio, or maybe all of those things happen at equal rates and therefore there's no difference in LC3 2-to-1 ratio. Maybe there's not even a difference in LC3-1 or LC3-2, and therefore no difference in the subsequent LC3 2-to-1 ratio because maybe, and this is entirely consistent with the mechanisms here, that the rate of LC3-1 production is exactly the same as LC3 1-to-2 lipidation, and the rate of LC3 1-to-2 lipidation is the same as the rate of LC3-2 degradation. Therefore, you don't have a change in LC3-1, you don't have a change in LC3-2, and you don't have a change in LC3 2-to-1 ratio. However, increased autophagy activity is occurring, and the same thing is true with P62. During increased autophagy, there's an increased expression. However, P62 is also degraded at the lysosome. Therefore, more P62 could indicate greater autophagy, or reduction in P62 can indicate greater autophagy. So when you go to measure these proteins, the amount that you're actually measuring is dependent on the relative rates of all of those different things. And another thing is the whole timing component. When were these measures actually taken? When were these samples actually taken? Because at one point in time, you may have more of a certain protein in the tissue, but if you were to take it later, say 1, 2, 3 hours later, you might have less of that protein because of greater degradation. And so how much protein is in that tissue when you go to measure it is dependent on when you took that sample, because maybe you caught it in the earlier phase or you caught it in the later phase. And the time course of these things, data suggest, are tissue specific. Now, there are other markers and proteins that can be used to indicate autophagy activity. However, you can have an increase in transcription factors or even proteins along the signaling pathway that don't necessarily translate into changes in a cellular process or system. So you can have an increase in autophagy transcription factors or an increase of an upstream regulator of autophagy, but that does not necessarily automatically translate to an effect on autophagy. It can give you more confidence with other markers. However, it's not so straightforward when you're looking and measuring autophagy in humans in real life. In cell culture and animal research, there are certain things that you can do to sort of tease those things out so you have a better idea of autophagy activity when you go to measure it. But in humans, in real life, in vivo, you can't do that. Now, I'm not saying you cannot measure autophagy in humans. All I'm saying is it is much more complicated, less cut and dry than it is often described on social media and on podcasts. With all that being said, in another study, 72 hours of fasting caused an increase of LC3 2-to-1 ratio and P62 expression in skeletal muscle of healthy young males. However, in contrast to the 36-hour study, in this study, instead of a decrease in P62, there was an increase in P62. So these two studies are a perfect example of autophagy being potentially indicated by different changes in some of these proteins. So it's not as simple as these proteins went up, so autophagy went up, or these proteins went down, so therefore autophagy went down. It's not so straightforward as that. And yes, having additional markers do give some further indication of what's actually going on with autophagy. But yes, in these two studies of fasting of 36 and 72 hours, including changes as early as 12 and 24 hours, autophagy markers, autophagy proteins did change in the skeletal muscle of healthy younger men. However, what you can't say is therefore 36 hours, 72-hour fasting is therefore good for your health, because they didn't look at long-term health. They looked at the effect of a single period of fasting, not long-term. It wasn't a long-term study looking at changes in long-term health. They specifically looked at autophagy in skeletal muscle in young, healthy men. And so they didn't look at autophagy in females, which there are data out there indicating that there are sex differences in autophagy between males and females. They also didn't look at individuals with chronic conditions like type 2 diabetes. They also didn't look at older adults. And that's not to throw shade at this research or these research groups. However, you got to keep in mind what you can actually say about these data, including how to generalize it. And currently, with these and any other relevant data, there are not enough to make autophagy prescriptions. There's not enough data to take and make practical applications and implementations into real practice. What you can say from these data is that time-restricted feeding will increase autophagy, protein, mRNA in whole blood in young healthy males or overweight healthy adults. And what you also can say is that 12, 24, 36, and 72 hours can increase autophagy in skeletal muscle of young healthy males. You can't make these broad generalized prescriptions that you should fast XYZ number of hours in order to turn on autophagy. Using intermittent fasting or time restricted feeding, TRF, or alternate day fasting, ADF, or 1 meal a day, OMAD, or LMFAO. You can't do that. You got to stick specific to what the data actually show. And what the data do show is that periods of fasting can increase autophagy. However, it's only shown in very, very specific contexts that should not be used for this broad, universal, generalized prescription. So the practical implications from these are somewhat limited. And again, you really can't make any sort of health or longevity claims off these data. So all these people talking all crazy online and social media about intermittent fasting or time restricted feeding, or again, 1 meal a day or whatever is anti-aging and going to live forever and things like that. I don't know what data they're looking at, if they are even bothering to look at data. Maybe they're looking at rodent data, which there are a fair amount of, but our biology and metabolism are very different. 1 meal a day is like 2 or 3 days for a human. A mouse will actually die in 2 or 3 days if it don't eat. Whereas humans, we can survive weeks without eating. So to look at something that was done or observed in a mouse and then take that and think that's going to translate directly to humans, like, you cannot do that. You cannot make that translation. It sounds nice. It sounds optimal. It sounds revolutionary. It sounds what the establishment doesn't want you to know so they could push big pharma. They don't know what they're talking about. And they're just using this stuff to push their supplements. But again, the people that are making these exaggerated claims about autophagy and longevity really don't know what the data actually say. And they're just saying it in order to push their content or their products. So for example, I came across this online. It talked about exercising in a fasted state increase autophagy to a greater degree than in a fed state. I have not heard of this paper. Let me check it out. But what they said was exercising in a fasted state increased autophagy more than in a fed state. And they went on to say that you should train in a fasted state in order to turn on autophagy more, in order to burn more fat, in order to presumably cause more fat loss. I said, that's interesting. And they include the reference. So that's nice. So I checked it out, and it turns out I had read this paper. And the reason I did not recognize it, because what this person said on this online thing was nothing close to what the paper actually did and what the paper actually concluded. This person completely mischaracterizes data, misrepresented the data to make it sound sexy. What did the study actually do? Well, they did look at exercise in a fed and fasted state, and they did see that autophagy was increased more. However, it wasn't in fat tissue like this person was presuming if they're talking about burning fat and fat loss. This was in skeletal muscle. And it was in mice, not humans. And on top of that, they didn't look at burning fat or fat loss. So 1, you got the model wrong. 2, you got the tissue wrong. And 3, you got the outcome wrong. That's crazy. But someone may come across this, read this, and say, oh, I'm going to start exercising in a fasted state in order to burn more fat, cause more fat loss because of autophagy. Because this study showed that, when the study did not show anything close to that. And the real question is, would any potential differences in autophagy between a fed and fasted state with exercise, would those differences actually matter? Would fed state actually matter? And again, for this, you would want to actually look at outcome data, not mechanistic data, not something that is going on inside of a cell, inside of a tissue. If you're interested in fat loss, in body composition improvements, and the exercise of how to do that, and whether or not exercising in a fed or fasted state is better for fat loss, you need to look at research that actually looked at whether or not the fed or fasted state had an impact on body composition and fat loss. Not autophagy studies, and definitely not in mice, and not in skeletal muscle. And what the outcome data actually show, which is the relevant thing to people in practice, is that there really are no differences in fat loss and body composition changes when exercising in a fed or fasted state. And it doesn't really seem to be that much of a difference in terms of the health outcomes between fed and fasted exercise. And that's the whole point. People coming across things because they want to figure out what they should do to get healthier, to improve body composition, and they come across something that completely mischaracterizes or misrepresents data, and then basing their practice on that, on bad or inappropriate data. And autophagy is a perfect example of this when talking about calorie restriction, intermittent fasting, and yes, exercise. Yes, autophagy is important. It's involved in health. It's involved in some of the benefits of exercise, like we'll talk about. And yeah, it may be involved in the positive benefits of calorie reduction and intermittent fasting. But to base practice how someone eats or trains on autophagy is just misguided. And this is an example of a larger point, but a main point of focusing on mechanisms. When mechanisms, from a practical standpoint, aren't that important. They're not the most important thing. What is the most important thing is getting to whatever outcome that you're trying to get to. So you should not look at mechanistic data in place of outcome data. And there are many reasons for this, but I think the most salient one is the fact that an outcome is not a product of one single mechanism. It is a product of many, many many mechanisms. And so there's no reason that you should focus on one single mechanism. So targeting one specific cellular process or some cellular phenomenon, it sounds cool, but most of the time people are actually doing things, training or dieting, because they have a specific goal. They have an outcome that they're after. And looking at mechanistic data, there's a very tenuous relationship between that mechanism and that outcome. And focusing on a single one, well, what about all the other ones? Those don't matter. And if we look at the outcome data of intermittent fasting, yeah, it does seem to improve health, including markers of age-related health. However, if you control for calories, there is no difference between intermittent fasting or TRF or 1 meal a day or whatever, between that and calorie restriction where you just eat regular. You just eat throughout the day. If calories are controlled, there is no difference in how those calories are consumed and when those calories are consumed. Intermittent fasting just may be a practical way that individuals can use to reduce their calories, which cool. If that happens to be an effective strategy for people to control their calories, then that's a good thing. But that's very different from it being the ultimate end-all be-all diet for health and lifespan and longevity and existential fears. The effect of exercise on overall health and age-related health is pretty clear. Like, there's nothing new about that. It does a tremendous amount in terms of improving overall health and preventing a ton of diseases, including age-related diseases, obesity, type 2 diabetes, neurodegenerative diseases, some cancers, psychological conditions like depression and anxiety, bone health, muscle health, liver health, pancreas health, heart health, brain health. Exercise almost literally does it all. The impact of exercise on overall human function, including the prevention of disease or even in the disease state of managing and treating and sometimes even reversing symptoms of disease, exercise has a tremendous effect. It is by far the best thing that you could possibly do for yourself. It doesn't prevent every disease, and nor does it reduce your risk of disease to zero, but it has a great effect. For example, VO2 max, which is a measure of maximum oxygen consumption during exercise, is the best biomarker for overall health and all-cause mortality. Better than any genetic test. It's an exercise test. We do them in our ex-phys labs like every day. Similarly, muscle strength, and to a lesser degree, muscle mass, the amount of muscle you have on your body, also plays a significant role in overall health and risk of disease and all-cause mortality. They say exercise is medicine, and for all intents and purposes, it is. And autophagy is involved with this process. However, it is not the only thing involved in that process. Like I mentioned earlier, people get super focused and hung up on specific mechanisms. Like, this is the thing that matters. This is a specific thing that needs to turn on when I exercise. And when in reality, like I said, biology ain't so cut and dry like that. Relating to this point, for example, not all exercise is going to turn on autophagy. However, that very same exercise will still promote positive benefits on health. So does autophagy really play a necessary role in the positive benefits of exercise? Based on the current research, and there was a recent meta-analysis that came out looking at this, for aerobic exercise, the duration of the exercise has to be at least 60 minutes and at 55% or more of your VO2 max or maximum exercise intensity. And with resistance training, though there's much less data on that, something like 8 sets at 10 repetitions of 70% of your 1RM seems to be required for autophagy increases. And with interval training, we really have no idea, because as of now, if it still stands, my dissertation study is the only study to have looked at high intensity interval training with autophagy in humans, in which we did 6 bouts of 1 minute maximal speed running at 3% grade, interspersed with 1 minute of walking at 3 miles an hour at a 3% grade, followed by a 5-minute recovery walk, followed by another 6 bouts of maximal speed running for 1 minute and a walk at 3 miles an hour 1 minute. It was pretty tough. And what we saw was a change in P62 in skeletal muscle following the interval training, but not any changes in white blood cells. And we also did notice sex difference. However, there's tons of data showing that exercise and just physical activity well below this autophagy threshold produces significant positive benefits on health and reduces the risk of many chronic degenerative diseases. 10-15 minutes of regular physical activity produces positive effects on health. There's even data showing that exercise for less than 5 minutes, albeit at very high intensity, reduces the rate of cancer incidence. So then that brings up the question of what is the actual role of autophagy in these positive effects? If exercise below what would be required to stimulate autophagy activity still produces positive health effects, then what is the actual role? Clearly, autophagy does not need to be acutely increased from exercise in order to produce positive benefits on health and age-related health, including those related to disease and premature death. In addition to that, there are data showing that exercise below the autophagy threshold is capable of leading to long-term changes in autophagy. 8 weeks of 15 to 20 minutes at 70 to 75% of maximal exercise intensity with some high-intensity intervals interspersed has been shown to increase some autophagy proteins in white blood cells in older adults. Now, the intensity is there, 70 to 75% of maximal exercise intensity. However, it's only 15 to 20 minutes. Yet there were changes in the actual proteins of autophagy in these cells. Also, 6 months of 150 minutes of walking or stationary cycling at an RPE or rating of perceived exertion of 13, alongside a couple of days of resistance training at an RPE of 15, led to changes in autophagy proteins in skeletal muscle in overweight women. Now, these women were in a 750 calorie deficit, so that definitely played a role, but we're still just talking about walking. And when we talk about long-term changes in autophagy, we don't even know what that means. So for example, there are data in mice, which are not human, so you can't take the data from these animals and then translate that into humans. But as we'll see, it gives us an idea that things aren't so cut and dry. There were 4 groups of mice. One group did aerobic training, the other group did resistance training, a third group did both aerobic and resistance training, and the fourth group, they were overtrained. These mice were beat up with exercise, and the long-term changes in autophagy were not what you might expect. It was the overtrained mice that saw increases in autophagy proteins in skeletal muscle, heart, and liver. And these mice were overtrained. They saw a decrement in performance, as well as other physical morphological impairment. And the trained mice saw decrements in autophagy proteins in heart, liver, and skeletal muscle, despite getting the positive benefits of exercise like the improved physical performance. And in addition to measuring the amount of these proteins, they actually also measured autophagy flux, or the amount of autophagy activity that's going on inside the tissues. And both trained and overtrained saw no changes in autophagy flux in the heart and liver. And there was an increase in autophagy flux from resistance training in skeletal muscle, while there was a decrement in autophagy flux with aerobic exercise and the overtrained group. Now again, you can't take these data and expect to see this exactly occur the same in humans. Can't do that. But what it does show us is we really don't know what to expect in terms of the long-term changes from things like exercise. We can't say more autophagy related proteins or more autophagy activity in the tissue is better for health and function, because in this study, there was an actual decrement in autophagy activity and no changes in autophagy proteins, even though in some of these trained groups, they saw the physical improvements from the exercise. And it was the overtrained group that saw increases in some of these proteins, which is a maladaptation. You can't say more autophagy proteins and more autophagy activity is necessarily better, that's going to be better for health and function, because maybe those things are elevated because there is more damage, because there is more stress, and stress in a maladaptive sense. And to cite some of the few relevant human data, when comparing endurance trained individuals to untrained individuals, there were some differences in some autophagy proteins, but no differences in others. And this was both fasted and after a high fat meal. And to cite some data from a study that I was a part of, While we did see a negative correlation between body fat percentage and LC3 2-to-1 ratio in white blood cells, we didn't see any sort of relationship between LC32 or P62. And when we compare these proteins, individuals that were above the 50th percentile and individuals that were below the 50th percentile, we did not see any differences in these proteins between these two groups. So this is all to say, we don't know what long-term changes actually mean or what we should actually expect from exercise, training, body composition, health status. We don't know what sort of autophagy composition would be reflective of those things. We don't know what sort of autophagy composition would be reflective of overall health or increased health or improved fitness. We don't know the composition or the level of autophagy activity is indicative, is reflective of actual improved health. And more importantly, from a practical applied standpoint, like trying to figure out what sort of exercise someone should do, that shouldn't even matter. What happens with autophagy shouldn't even matter. What matters is actual achieving your specified outcomes, achieving your goals. If you're someone who's trying to exercise or eat in order to improve your health, like reduce your type 2 diabetes risk or increase your insulin sensitivity or improve your blood lipids or reduce the risk of heart attack, things like that, what happens with autophagy shouldn't even matter. What happens in the cells with autophagy or anything else really isn't important. What's important is getting to those goals, figuring out how to achieve those goals, and identifying and targeting a specific mechanism is not the way to do that. It's just missing the point, because autophagy might be involved in that outcome, but it is not the only thing involved in that outcome. And to reiterate, there's exercise that is not going to upregulate autophagy that is going to have huge positive impacts on your health, like type 2 diabetes risk, insulin sensitivity, blood lipids, and heart attack risk. And likewise, exercise or physical activity that is not going to acutely increase autophagy activity has been shown to produce long-term changes in autophagy. And what do those changes even mean? We don't know what rate or changes in rate of autophagy actually has any sort of health or physiological impact. What amount will affect cellular function? What amount will actually affect health? Does 10% have an impact? Does 20%? Does 1%? We may look at a study and see that there was not a statistically significant increase in autophagy. However, that increase may be actually meaningful from a physiological standpoint. That change might actually affect function. That change might actually be physiologically relevant, but not statistically relevant. Similarly, we may look at a paper and see, oh, there was a statistically significant increase in autophagy, but that may not be physiologically meaningful. Statistically might be different, but that difference or that change may not actually mean anything from a physiological, functional, clinical standpoint. That statistically significant increase may not actually mean anything because we don't know what level of autophagy or changes in rates of autophagy actually have an effect on function or on health. Even if autophagy was worth targeting, that you should train and eat trying to increase autophagy, you can't measure it. Like, you can't measure it. I've measured it. It ain't easy. There is no readily available way to measure autophagy via some app or even some laboratory testing. You really just can't go to some wellness or fitness center or holistic medicine thing or your doctor or even a lab and say, measure autophagy in me. And if that were the case, the follow-up question would be, okay, in what tissue? And you don't know. What tissue are you actually talking about? They haven't thought beyond turn on autophagy good. Measurements of autophagy have to come from some tissue. They have to come from some cells, meaning you have to take tissue out of the body. You have to take cells out of the body. If you want to measure autophagy in skeletal muscle, guess what? You got to give up some skeletal muscle. You got to get a biopsy done. That's literally taking a chunk of muscle out of your body.
Good. Okay. So now, next step, tissue, and then you're going to set the gun to the side.
Okay, so this, I'm not going to touch it.
And I could put it in any orientation that you want.
That's
to
great.
center.
Yeah.
And we're already done, so we could actually touch it, right, if we needed to.
If you want autophagy measured in fat cells, well, guess what? Then you got to give up some of your fat cells. They got to go in and take a chunk of fat cells out. If you want autophagy measured in your liver, they got to go take a chunk of your liver. Pancreas, same thing. Brain, same thing. And I will say you can get autophagy measured in some cells from a blood draw. That's something that I've done a lot of. And that's coming from white blood cells. But again, that's not a readily available or commercially available option. So if your goal is to focus on autophagy through exercise or intermittent fasting or calorie restriction, there's no way to even know if that's having any effect. You cannot measure that. You don't have access to a measurement of it. So focusing on autophagy doesn't even make sense because you can't measure it. You don't know what you're doing is actually having an effect on autophagy. And like I said earlier, if there is an effect, we don't even know what those effects mean. So trying to do something to upregulate autophagy, you don't even know if you're upregulating it or not. And this point right here illustrates the irrelevance of focusing on mechanisms in practice, focusing on mechanisms in your training or in your nutrition. You can't measure most of these things. You're not measuring the mechanisms. So for example, when people talk about, say, exercise and mitochondrial biogenesis, you're going to exercise and that's going to cause mitochondrial biogenesis, making more mitochondria, good functioning mitochondria, that you should target your training for mitochondrial biogenesis. These things have to be done in a lab with specialized equipment and supplies. So targeting mitochondrial biogenesis with your exercise doesn't make any sense because you're not going to be able to measure, did you make more mitochondria? There is no app or scan that will tell you you made 5 more mitochondria. Instead, you should focus your training on the actual outcome that you are trying to achieve. So if you're interested in increasing your, say, aerobic fitness, then you can, go have your VO2 max tested and see whether or not did you get more aerobically fit. And guess what? If your VO2 max increased, chances are that you made more mitochondria. You did have more mitochondrial biogenesis. That increase in VO2 max would highly suggest that you have more mitochondria in that muscle. Or even easier, and because not everyone has access to getting a VO2 max test done, that costs money, can you just run for a longer period of time without getting tired? Can you run harder for a longer period of time? Can you bike or swim or whatever your training is? Can you do that for a longer period of time or do that at a greater intensity for a longer period of time? And if the answer is yes, then guess what? You probably made more mitochondria. So if your goal is to get healthier and you've heard about the importance of mitochondrial dysfunction and mitochondrial biogenesis, or even say activating GLUT4, which GLUT4 are the transporters that allow sugar, glucose, out of the bloodstream into the skeletal muscle. And exercise increases the activity of GLUT4, causing blood sugar, blood glucose to decrease acutely, as well as lowering overall blood sugar levels, blood glucose levels, long-term, improving glucose regulation and lowering blood glucose levels. And that includes making more GLUT4 proteins. So similarly, exercising to try to activate GLUT4 doesn't make any sense because you can't measure GLUT4. You're not measuring it. But guess what? If your blood sugar levels improve, both in the short term, like post-exercise and 1 or 2 days after exercise, as well as just baseline blood glucose levels, if they've come down, if your A1C has come down, that is highly suggestive that you, yes, activated and made more GLUT4 transporters. But you can't measure it. So don't even consider trying to target GLUT4. Target trying to improve your blood glucose levels. And if you're not insulin resistant or type 2 diabetic or pre-diabetic, or your A1C is in good healthy range, or your fasting blood glucose is in good healthy range, there's no reason for you to be using a glucose monitor. If you're someone with insulin resistance and diabetes, then yeah, you do need to check your glucose. That's important to monitor. But if you're not someone that has insulin resistance or type 1 or type 2 diabetes, has a healthy A1C, has a healthy fasting blood glucose, you don't need that. You don't need to monitor your glucose post-exercise or after every meal, you're fine. So if you're someone that wants to improve their health and prevent their risk of disease or improve their symptomology of disease, and want to do that through exercise and nutrition, and then you get online or social media and Instagram or TikTok, and you see a bunch of people talking about autophagy and GLUT4 and mitochondrial biogenesis, all of these things you cannot measure. Focusing on them does not make any sense. Well, this type of exercise has been shown to increase mitochondrial biogenesis, or this type of exercise has been shown to increase autophagy. Cool, that's great, but that's not reducing your A1C. That's not reducing your blood pressure. That's not improving your blood lipids. That's not reducing your C-reactive protein. If you're trying to improve your health, then you're actually trying to improve your health. So instead of looking at research or consuming content on Instagram and TikTok and YouTube and podcasts, talking about this exercise increases autophagy, this exercise increases mitochondrial biogenesis and GLUT4, those studies most of the time are just showing that it had an effect on this cellular thing, that it had effect on this mechanism. That's not the same thing as an outcome. That's not the same thing as reducing your A1C, improving your blood lipids, reducing your blood pressure. That's not the same thing. So you should be looking at research or considering content that talks about this type of exercise has been shown to reduce A1C, improve blood glucose, improve blood pressure, decrease C-reactive protein, not some single mechanism in a cell. If you're someone who is trying to improve their health through training and like nutrition, and after some time your health improves, well, guess what? Autophagy, mitochondrial biogenesis, GLUT4, they were probably involved with that. But guess what? You didn't need to know or measure those things in order to improve your health. If you improved your health, you lost weight, your blood pressure got better, your blood glucose got better, guess what? You're healthier. If those things were involved, then they were involved. If they weren't, then whatever. Now, they probably were to some degree, but focusing on them and targeting them and highlighting them is not relevant. Focus on the things that you can measure and measure the actual things that are going to affect your health and your goals. How many posts or thumbnails do you see that say something like increase X by 400% or never do insert bad thing here again? Like do these 3 simple biohacks or this one secret trick. Full tricks are for kids. Like that's not science, that's marketing. And like something like this, talking about working at the cellular level, which all that we are ourselves. So if something works, by definition, it works at the cellular level. Good luck getting past the cellular level. But it sounds sexy and people are going to gravitate towards it. And I understand not everyone is a scientist. Most people aren't scientists, but that's kind of the point, because most people don't have a background understanding of certain things where they can read a research paper or look at data or listen to something online that talks about something sciencey, something research based. And they don't have the ability to really interpret that data or that information that is being presented in a way that is actually representative and appropriate to that data. Like people consuming that content, they don't know. They don't have a background in these subject areas to be able to discern those types of things. And so there are issues with people coming across this content, trying to figure out ways to improve themselves and make themselves healthier and get more fit and all of that, and adopting these strategies or protocols when there is actually very, very little data to support their use. And understand that talking about these things can get people more interested in their health. That could lead to positive behavior changes. But the issue is people are being misled and they're buying into things that are oversold and oftentimes don't have data behind them. A lot of times what they're doing is they're taking mechanistic data that sounds super fancy and novel, which it is really interesting research. But what they do is they take that and translate that into context of which you cannot do, including informing how humans should train or eat and confusing mechanisms and outcomes. It grabs your attention. It sounds super sciencey, like frontier of science type stuff, but they're really just using that language to get you to consume their content or buy their program or download their app or buy their supplement. That's the game they be playing a lot of the time. Misrepresented data be damned. And a lot of times I think individuals just take their information based on vibes. And science has become a vibe. It's an aesthetic. It's a marketing tool. Science, or the aesthetic or vibes of science, is now just a tool as a commercial for products, for content, for services, most of them unnecessary and or lack any sort of empirical evidence to them. And there's nothing wrong with talking about interesting science and research, including stuff that has like big time implications in the future potentially. But there's a difference between talking about the specific data and the potential applications or impact, sometime in the future, versus talking about these really obscure, esoteric, basic data as if they have application now in real life for people to take and use, for people to apply. Now, I'm not saying terms like backed by science or science-based are bad things. If I'm working with a client, I'm gonna do things that are science-based or evidence-based, backed by research. That's not the issue. The issue is when those terms or science or research are used unscrupulously in order to push something. And the idea of the science or being science-backed is used more as a marketing tactic used by people or companies or people on behalf of companies. It's to create a vibe, to get people to want to engage or buy your stuff, to create the feeling of, oh, this is science-backed. I love science, where science is primarily a means to drive revenue, rather than the science itself being used to validate the claims that are being made and the efficacy of the use of whatever the product or service is. It's an aesthetic more than it is a principle. And most of the time, if you go track down the science that is being cited, it's oftentimes very different than the claims that are being made, including times where the data do not substantiate the claims at all. These claims are based on very, very shaky data or have a very, very weak and tenuous relationship to the data they're actually citing. And they're really not that science-backed after all. There's nothing wrong with talking about this science. There's nothing wrong with talking about these data. This stuff is really cool. It's one of the reasons why I became a professor of exercise physiology. So I could talk and study these things. Most of my classes are like 3 hours long, and that's never enough time. I have to force myself to stop talking about these things. And I never get through all the things that I want to get through in a semester. So I understand talking about these topics and this research is really interesting. It is really cool. But you have to do so with all of the necessary and appropriate context related to that data, so they don't come away with the wrong impression or idea of their significance and actual direct impact to them. There are so many things on social media and podcasts and like thumbnails that, as a scientist, someone who actively does research, I would not feel comfortable saying. There are things as a professor, as an educator, that I would not feel comfortable saying to my students in that way. And there are things that I would not feel comfortable saying as a practitioner if I was talking to one of my clients. However, there are scientists and doctors and other professionals that are making statements or presenting things that are just unfounded, misleading, and especially if you're someone that is in science or in medicine or in higher education, that are just disingenuous. If you take some of these individuals out of the podcast TikTok space and actually put them in their professional setting where there are standards of practice, where there are ethical norms or legal implications, whether that be in a classroom with their students or in a lab with their research group or a physician with a patient, like they would not be saying the types of things that they are saying on podcasts to those people in those settings. And I understand if you're talking to a general population, you can't talk as if you were writing a research paper or at a professional scientific conference. But as a scientist or an educator or a physician, you at least have to be committed to the actual data. This is what I have to do with students. You have to communicate things in a way that it is understandable to them, but you still have to reflect what the actual data and research say. There's a difference between education, including passionate education, and like proselytizing, like you're some preacher or TV evangelist. A student asked me, after giving like a 2.5-hour lecture on autophagy and finishing up with the conclusion here of like autophagy from a practical standpoint really is not that important, she asked, well, if it's not important, why do you study it? I said, that's a good question. That's a fair question. Not all science, not all research has direct application to your life. Not all science is translatable into action, and that's okay. Sometimes we just need to know things for the sake of knowing things. And maybe sometime down the line, then there could be applications made. But not all science has to have direct applications or implications. Autophagy does have potential applications and implications, which I'd be interested in figuring out. But to be honest with you, I just think it's interesting. I think how the body responds to the stress of exercise is interesting. I think the fact that this system is billions of years old and we share it with really simple organisms like yeast tells us how important it actually is because it's been conserved over billions of years. I think that's really interesting. And if there are applications that can be made, well, I'd be interested in finding those too. And people can research different things. People can research things that are really, really applied, or people can research things that are really, really basic and have little to no application. And there's a whole area between those two ends of the spectrum, of which exercise physiology is making ground in. Now, this is all as of early summer of 2024. And new data can come out. Things can change. New research can emerge. And for sure, there will be. In terms of these data and the interpretation of these data, I'm much more comfortable with maybe, potentially, likely, possibly, implicated, associated, those types of terms. Not these definitive, conclusionary, absolute statements, but these terms lack certainty. But guess what? A lot of things lack certainty. I understand that someone can look at this data and come away with a different interpretation and say, I'm perhaps being too stringent. I perhaps am being too conservative when it comes to interpretation and translating and applying what the current body of evidence is. And yes, I am being conservative and stringent, especially when it comes to taking specific data and then applying them into a context outside of what that study was performed in, particularly of taking these data and then applying them into practice. I myself am active in research as well as a practitioner in the field. And as a researcher, I have to contend and consider what the limitations of my research is. What can I actually say and conclude from my data? Where and when might it matter? And when does it not? And trying to not overstep that line. As a researcher, I have to consider, does this even matter? And be okay with whatever the answer is. And as a practitioner, I have to read the research and try to discern what actually matters in practice, what is actually relevant and what is not. And in practice, I don't think about autophagy at all. In total, don't focus on mechanisms. They're interesting, they're important, but they have very little relevance in terms of application and practice. Do things that are effective at getting you to your goals. Train for your goals, eat for your goals, and use research and literature that are related to reaching your goals, rather than on a mechanism. Because outcomes are not a function of one single mechanism, but they're a function of many, many mechanisms. And if that mechanism is involved, like autophagy, then it's going to be involved as much as it needs to be. It doesn't need to be targeted. It doesn't need to be focused on. It's going to be involved as much as it needs to be. And don't get caught up on the sciencey sounding, pseudo-profound, overly complicated, biological, biohack, master this, cure that, optimize, maximize, never do XYZ again, because there's a lot of shit online and on social media, and a lot of it ain't science, it's vibes and commercials. If you like this content and want more on exercise physiology, training, and nutrition, as well as interviews with researchers and other practitioners in the field, support it on Patreon, where you can get exclusive content. But for now,